Hello viewers, today I am lecturing on a new topic. I hope everybody will get interest. The topic is seedless vegetable and fruit productions by genetic engineering technology. We know here there is seeded fruit and seedless fruit. So today my topic is on seedless production. The details topic is seedless vegetable and fruit productions by genetic engineering technology. It means genetically modified organisms using hormones J3IA hormones and regulated gene expressions. Importance of this study or introduction. Fruit size development is an important issue by genetic engineering. Breeding of plants is one of the significant methods to get expected results such as small seedlets or bigger size fruits. Has been done for a long time. In 1956, in 1856, Mendel, the father of genetics, we know he was the father of genetics, first published his findings on the statistical laws governing the transmission of plant traits between generations. Mendel studied how a specific trait in the peas plant in his garden had passed down to succeeding generation, and he formulated the idea of some sort of unit within plants that was responsible for heredity. In the 20th century, researchers had established that inheritance is transmitted by gene. This is Mendel's theory, which expresses chemical information resulting in characteristic trait. For seedless fruit, it is important to understand more of the details of genetic transmissions. Genes in plants and animals are usually deployed in pairs called an allele. One gene in the allele is usually dominant and the other recessive. There is two genes. One gene is dominant, another one is called recessive. This means typically only one trait is expressed in the biological makeup of the organisms, though there is still a second gene for the traits. This is an important because every cell is an organism carries a complete genetic makeup of itself called the chromosomes in its nucleus. There is a nucleus in the chromosome. There is a nucleus. There is a chromosome uh, inside the nucleus. When a cell divides, the chromosomes make doubles, and then a copy enter into the new cells. The exceptions is the sex cells, the ovum and sperm. There is two cells, we can say ovum cells and sperm cells. For the females, we can say ovum, and for the males, we can say sperm. These cells only carry half the genetic materials, which is one chromosomes or one half of each gene pair. When the ovum and sperm meet, the gene pairs recombines and the new individuals created through sexual reproductions has a new full set of genetic materials, having half inherited from his parents. So Mendel, he invented this in case of plants, but now this theory is now we are applying also in animals, in men, in human cells. Genetically modified organisms GMO technology keeps a significant role to produce GM organisms or seedless and GM genetically modified organism, dry fruits or seedless. GM or seedless is generated by genetic engineering technology. This is the same protocol, the the genetic engineering technology. We can produce GMO and seedless. The first GMOs are bacteria in 1973 and GM mice were generated in 1974. Insulin producing bacteria were commercialized in 1982 and genetically modified food has been sold since 1994. Jam is the source of plant, animals, medicine, and genetically modified foods, also widely used in scientific research and to produce other goods. So we can see here seedless orange and grapes. Now we can see in the market seedless orange and grapes. So at the result of cultivation of the naturally occurring seedless plants, the Naples orange is descended from a seedless orange tree plantation found in Brazil in the 19th century. This tree was the result of mutation breeding. This is called the mutation breeding. That is, something in the genetic materials had spontaneously changed, resulting in these unique plants. Orange growers propagate new trees from the original level. Seedless grapes. Now you can see also seedless grapes in the market. Seedless grapes, even red and black varieties, are grown in USA by Thompson's until 213. The Thompson's has a genetic abnormality that causes the seeds to arrest the development. Though the flower is pollinated and to ovum fertilized. This is the technique, plant breeding techniques by pollination. You can see the protocol or technology. The flower is pollinated and the ovum fertilized. The seeds stopped growing after a few weeks. So the grapes is not entirely seedless. Is not entirely seedless. Rather, the seeds are aborted. Some seeds aborted, maybe 5%, 10%, 20%. That is seed. And exist tiny specks inside the fruits. Commercial growers treat the plants with a growth hormone called gibberellin, which is normally secreted by developing seeds. The flowers are dipped or sprayed with the hormones so that the grapes grow big and juicy despite the arrested seeds. The technique is the dipping method, and they use the gibberellin to make the seedless. So this is seedless watermelon. Now we can see in the market began in United States markets in the 1990s. Seedless watermelon is sterile, that is seedless. Because they have 3 sets of chromosomes, the condition is called triploid. The standard watermelon, like Thompson's grapes and most other organisms, have 2 sets of chromosomes and are called diploid. To produce the triploid watermelon cell, diploid parents is pollinated by tetraploid watermelons, which has 4 chromosomes number. During sexual reproduction, the new chromosome inherits half of this parent's genetic materials. As a result, the new watermelon gets 1 chromosome from the diploid parents and 2 from the tetraploid, making it triploid. So the triploid hybrid is virtually seedless. It produces very few seeds, and this can be planted to grow new watermelons. But the new plants must be pollinated by standard diploid watermelons in order to produce fruits. The development of a new strain of seedless fruits or vegetables is a stocking process. This is the different process. Research is typically carried out by horticulturists working at an agricultural development laboratory or governmental research station in different countries, especially in USA, where they can develop years to the work. A researcher studies thousands of seedlings to find ones with the desired characteristics. So there's many techniques. First techniques, we can do seedless by breeding. Plant breeding, seedless by first is plant breeding by pollinations, and then cross breeding, seedless by cross breeding. We can say this is the different techniques. Then seedless fruits and vegetables by using growth regulators or hormones. This is by mutation breedings. And others we can see here parthenocarpic fruits and vegetables, another techniques, parthenocarpic techniques. And then others we can see here seedless vegetables with physiological, this is called the physiologically changed. So this is the by environmental factors, physical change by environmental factors. So we can now discuss one by one. One is, we can see here other materials and methods. Seedless by crossbreeding. The traditional process for breeding seedless fruits was to cross a seeded female plants with a strains of seedless male and female. The offspring was seedless about 15% of the time. Then a successive generation could be produced from this 15%, starting in the 1980s. One scientist, he developed this protocol. Horticulturists found ways to speed up the process by culturing the tissue of seedless plants with grapes. The aborted seeds of the seedless strains are grown in the petri dish or test tube. Then this seedless strain can be crossed with other seedless strains, resulting in offspring that is found 50 to 100% seedless. So this is one by one in vitro culture. This technique has been used with great success with grapes, speeding up the time it makes to bring a new seedless variety to market as commercially. With watermelons, the sprouting tips of a seedless plant is placed in a petri dish filled with growth regulators and nutrients, and the one tips would sprout as many as 15 grown plants. This technique has also been used to produce seedless tomatoes. This is one kind of techniques. Other technologies, seedless fruits and vegetables by growth regulators by hormones, making mutation breeding. Sudden change of gene. This is called mutation breeding. Many of the fruits produced in this way are seeded. The greatest benefit is that tomato crops can be grown during periods of cloudy weather and low light intensity. And all year round we can produce vegetables. Now we can see there is some vegetables in summer, vegetables and winter vegetables. So by this technique we can running this process now, growing of the vegetables all year round, summer vegetables and winter vegetables, and also summer and winter vegetables. There is no restriction. All year round we can produce the vegetable and fruits. And during the short days of winter and the early spring, sprays with the beta naphthoxyacetic acid and alpha orthochlorophenoxyacetic acid. This is called a different type of organic plant growth regulators. And parachlorophenoxyacetic acid and others have been used effectively by commercially grow of greenhouse tomatoes. In greenhouse tomatoes we can use this. If the flower cluster are sprayed just before pollen is shed, most of the fruits will be entered or almost seedless. The yield of this field-grown tomatoes, particularly in the first picking, has been increased by the use of these regulators, notably when crops bloom under conditions of the low night temperature and are unfavorable for fruit set. In 2012, we reported that gibberellic acid, inbutyl acetic acid, and naphthenic acetic acid produce seedless okra at different concentration by using stem, flower, and over injections before flowering. Weber, 1969, also discovered the gibberellic acid is superior to other growth regulators. He tested as an aid in increasing the size of the grapes, particularly in the Thompson seedless variety. So, other techniques, parthenotherapy, fruits and vegetables. Aborted seed, that is aborted seed. Most of the fruits and vegetables started growing after pollinations when the pollen sperm has fertilized the plant's ovular compartment in its flower. Plants which grow flower without pollinations and thus have no seeds are said to be parthenotherapy. This is called the parthenotherapy. Plants which grow fruit without pollinations, this is called the and there is no seeds, are said to be parthenotherapy, from Greek meaning virgin fruits. This is called the virgin fruits. Fruits bearing plants all grows parthenotherapy fruits, which usually differ in size and appearance than the regular fertilized fruit. Some fruits are naturally parthenotherapy and don't need any interventions to produce many seedless fruits. Bananas, for instance, are triploid, 3 chromosomes, and rarely produce eggs or sperm with the very chromosomal layout for reproduction. Seedless watermelons works much the same way, only they are not naturally triploid but must be bred from a diploid, 2 chromosomes, and tetraploid, 4 chromosomes parents, which itself must be manipulated to be tetraploid. Clementines and the Mabels, one scientist, Clementines and the Mabels orange, that is the name of the orange, has itself incompatibility genes that keeps flowers from being pollinated by the same plants. If every tree in the orchard is cloned from one plant, all of the pollen is exactly alike and appears to the flowers to be from the same plants. When that happens, the trees grow their fruits parthenotherapically, and seedless orange are the result. Scientists have also discovered that the hormones auxin can cause normally non-parthenotherapy plants to produce fruit without being fertilized. Auxin usually encourages the plant to grow shoots in one direction or another, but when exposed to the ovary, it lets the fruits begin growing without pollinations. This discovery hasn't been brought commercially yet. Seedless vegetables, pumpkin and lettuce finger with the okra or bhendi produced by hormone treatment. GA3. Pumpkin plants, local cultivar were grown at the different experimental field, and then the concentration of 150 ppm in GA3 was used for 5 replicants and with water control. Here we use the injection method by using microsyringe method, and then we make the seedless pumpkin or reduce seed by flowering before blossoming. We can see here, this is seedless pumpkin. This is seeded pumpkin. When seeded pumpkin after application treatment, this is the seedless pumpkin. My PhD student did this work in Malaysia. Photo shows the seedless and the seeded pumpkins by genetic engineering topic. So this is when you use the hormone and sudden change of the gene. This is belongs to changing of the gene. That why it happens. Seedless star fruits productions by hormone treatments. We use J3 150 ppm treatments in the trees, and then you can see by which flower bud were dipped twice per week for 2 weeks. Flower bud dipping method in J3 150 ppm during the growing season, the flower bud formations. And this technique without pollinations. It belongs to mutation breeding. Sudden change of the gene of genetic materials. We can see here this is the seedless. This is seeded and this is seedless. This is seed. There is seed. There is no seed. Seedless. There is seedless star fruit productions. We did my graduate student this experiment in Malaysia. So, and also you can see here seedless okra, ladies finger, or the Mendel by IA. My PhD student this work 1.5 ml, 1.5 ml of the concentration of 1 IA 100 milligram per liter ppm, but applied on the okras flowers by injecting once with needle before anthesis, while control was distilled water. That is distilled water. And green pod length and pod diameters, healthy pods, seedless and aborted seed are measured. You can see here the flower before injections, flower bars and flower injected, and then this is the seedless okra, reddish finger, and this is the without seedless. There is no seeds. By the genetic engineering techniques on mutation breeding, we produce this, but commercially it is not yet coming in the market. So that is still under research. We can see here now result and discussion. Pumpkin weight and seeds number measurement. We can see here the J3 and control. All seedless, 96.9% seedless, and then seed weight, fruit weight, seed number, also here seed weight and milligram, and then also here fruit weight is little bit higher than control. Here we can see biochemical contents, glucose, fructose, potassium and calcium, all are almost higher in the J3 their control treatment. Now you can see here table 1 already discussed here, where you can see the others. For star fruit, you can see here the water control and the fruit weight, seed number, seedless fruit and fructose and seedless, 75% seedless. And then here we can see fruit weight also higher in case of J3, and the fructose also higher in J3 than water control. So it has been shown that fruits weights are higher in J3 treated fruit than in control. However, seed number are higher in the control than the J3 treated the fruits. Besides, 75% seedless aborted seed star fruits are found by treatments of J3 compared to the control. Okay, let's see to other experiment. Here you can see okra or lettuce finger, fold length and the biochemical contents. Fold length, total soluble solid vitamin C, seeded pods and seedless, 100% seedless. In case of lettuce finger, we got, and then vitamin C also higher, and then fold length, TSS content higher in IAA 200 ppm, then water control. In case of okra, lettuce finger, so you can see here there is some little, okay, the maximum pod length, TSS and the vitamin C were obtained in IAA 200 milligram per liter compared to the control. The significant highest seedless pod was 100% in IAA to 200 pp. milligram per liter, followed by 0 in control 200 milligram per liter. However, seeded pods was 84.3% in the control, followed by the 0 in the IA treated pods. From the results, it has been seen that maximum seedless pod was 100%. It might be due to the genetically modified, means by IA hormones. It means the mutation breeding happened. It has been reported that Pericarp PSGA3 OX1 express was formally regulated, and the conversion of the J20 to J1. Actually, gene character changed. This is called a genetic engineering. The gene makeup, gene, the structure of the gene means the characters of the gene has been changed, regulated by the presence of seeds and the forced CLIA for food growth. You can see here list of genetically modified seedless fruits and vegetables, Joseph 2013, and to the different. This is the website. You can see here this is our seedless okra, seedless pumpkin. We published this paper in Scientific Horticulture, very high impact factor, Science Direct, American journal, impact factor 2.87. So also others, you can see seedless watermelon. Now in the market you can see in the seedless pumpkin also. Nowadays in different countries you can see, but and there is a seedless watermelon, water apple, seedless persimmon. In Japan also it is very famous. Seedless pineapple, seedless lemon, seedless orange, seedless grapefruits, seedless tomato. Now we are eating seedless papaya, seedless peach fruit. And then here also seedless pear, seedless loquat, seedless palm, seedless guava, seedless grapes, seedless apples, seedless banana, seedless pig fruits, seedless eggplants, brinjal, and seedless cucumber. Then seedless cumis melon, this melon, which is called another type of melon. And then seedless cactus pears also you can see here, and seedless blackberry. So different types of the fruits now we are getting in the market, and this seedless. And then this seedless, actually this is these seedless are very important nowadays. And what we are doing, we are saying about hybrid regulation to gene expression. Why we are getting this type of fruits seedless? Because there is changing of gene. That's how you can see here auxin and IA regulated gene expressions. When you use the hormone, then it changes the gene characters. J3 regulated gene are expressed in Q beta maxima by northern blot. This is the protocol. Northern found new genes. The CMG J3 OXN gene changed. Also others we can see here gibberellins, and they call gibberellin biosynthesis in P. And then they observe other scientists get the gene also changes. And this is from gene bank. There is a bank, gene bank. We analyze this, and then we can see this type of gene code for the enzymes and the converted biologically actives gene from G20 to G21 GL. Also there is another type of PSGA30X1 mRNA level, where minimally detectable in free-pollinated pericarps and ovules, and different types of treatments. So also here there's different types of gene characters developed. The data showed that this type of genes was expressed and developed and regulated in the pericarps and seeds. So this type of, we can see here also the expression of the GA3 regulated gene in Q beta maxima. Here GA3 they are controlled. There is no expression, there is no genes here, but here after application of GA3 the gene expressed here. Here you can see a DNA band. So this is the expression of gene, and what type of gene is this, and we can find it from the gene bank. Scientists got it. Then I regulated gene expressions. Also, I regulated gene expressions. There's the type of gene expressions we can find in different AFR2A in tomatoes. Different types AFR2A in the tomatoes. AFR2A auxin and IA regulated gene. One is main gene, original gene, and then after that, when we use the treatment or hormones or any kinds of the treatments, then it changes this gene characters, and new gene expressed. Overexpressing fruits displayed and ethylene emissions and ethylene signal inhibited delayed their ripening phenotypes, suggesting ethylene dependency. And then different types of comprehensive hormone profiling revealed that altered AFR2A expression in fruits significantly modified abscissions, cytokinin and salicylic acids. Different types of auxins, gibberellin and auxin metabolites are unaffected. So, in conclusion or suggestion, you can say from the above results, it can be concluded that genetic engineering technology can be applied using hormonal injection technologies, solving in xylem, phloem, some also technology exhibited or bud or before flowering or after flowering, exhibited an innovative in pumpkin, lettuce, finger, and the star fruits seedless. This is called also GMO technology, genetically modified. Therefore, GMO plants and fruits and vegetables can be produced by using different concentrations of GA3, IA, IA. This is the, and also in others experiments, it can be conducted that 96.9% seedless pumpkin was produced at GA3 150 ppm. So besides GA3 150 ppm was the best treatment compared to the showing the 75% synthetic aborted seeds. So in others, you can see here aborted seed, the aborted seed and biggest size and the highest TSS and biochemical content of foods. In addition to the nutrient contents, sodium, calcium, potassium higher in GA3 150. In case of GA3, in case of okra, 200 ppm and 100 ppm the best for, because it became 100% seedless. So this is the references for the different scientist name. We publish also many papers on seedless. So in different journal, American journal, European journal, and there this is our name here. So also we publish the books. Also there is books, you can find the plant physiology and biotechnology, principles of biotechnology, plant biotechnology, genetic engineering, 3 books, and book chapter also you can find. When you search my name in the Google, you can find my names and the links. And thank you very much for listening, watching, and subscribing our channel. Thank you again.